Hello friends, in this video we are going to see about the biosynthesis of peptidoglycan, especially in gram-positive and gram-negative bacterial cell wall. So this is John Avinash who will be presenting to you the whole of this video. First, let us locate where the cell wall is. We all know that every cell is made up of the cell wall, especially the bacterial cell is made up of cell wall, and below that there is the plasma membrane. The bacteria may be conveniently divided into two further groups depending upon their ability to retain a crystal violet iodine dye complex when cells are treated with acetone or alcohol. This reaction is referred to as the Gram reaction, which was named after the scientist Christian Gram, who developed the staining process or the protocol in 1884. There are mainly two types of Gram staining. First is the Gram positive and the second the Gram negative staining. The definition of peptidoglycan. Peptidoglycan, also called as murin, is a polymer that makes up the cell wall of most bacteria. It is made up of sugars and amino acids, and when many molecules of peptidoglycan join together, they form an orderly crystal lattice structure. You can see the lattice structure here of the peptidoglycan. Bacteria are classified as being either gram-positive or gram-negative bacteria based in difference in the structure of the peptidoglycan cell wall. Archaea do not have a cell wall containing peptidoglycan, but some do have a layer of pseudopeptidoglycan, that is pseudomurin, which is a similar polymer. Let us locate where the peptidoglycan is present in both the cell walls. Here you can see in the gram-positive cell wall, the peptidoglycan is present above the plasma membrane. Whereas in gram-negative bacteria, you see the plasma or the plasma membrane is below, and above that there is a plasmic space, periplasmic space, and above that there is the peptidoglycan which is below the outer membrane. The cell envelope of prokaryote. Gram-positive bacteria and gram-negative bacteria differ in the appearance of cell wall. The cell wall of gram-negative bacteria is multi-layered structure and quite complex. Gram-positive bacteria contain primarily single type of molecule and is often much thicker. You can see here the two types of cell walls and the peptidoglycan cell wall or the peptidoglycan part in the cell wall. In gram-positive bacterial cell wall, as I told you, it is present above this plasma membrane, whereas in gram-negative bacteria, you see it is present as a sandwich in between the outer lipid membrane and the plasma membrane. Here we need to know about the three different bonds or the three different links. First, the pentaglycine cross-link. Second one is the tetrapeptide link. And the third one is the NAG or NAB link. Okay, that's the three thing we need to understand here. First, bacterial cell wall and peptidoglycan component. In the cell wall of bacteria, there is one rigid layer that is primarily responsible for strength of the wall. In gram-negative bacteria, additional layer is present outside the rigid layer. The rigid layer of gram-negative bacteria and gram-positive bacteria is very similar in chemical composition and is called as peptidoglycan or murin. This layer is thin sheet composed of two sugar derivatives, that is NAG and NAG, NAG and NAM. The N-acetylglucosamine and N-acetylmuramic acid are the two sugar derivatives, and small number of amino acids consisting L-alanine, D-alanine, D-glutamic acid, and either lysine or mesodiaminophilic acid. So these are the two different components which are mainly present in the peptidoglycan. These constituents are connected to form a repeating structure glycan tetrapeptide. And basic structure of peptidoglycan is a thin sheet in which the glycan chains formed by sugars are connected by peptide crosslinks formed by the amino acid. Glycosidic bonds connecting the sugar in the glycan chain are very strong, but these chains alone cannot provide rigidity in all directions. In gram-negative bacteria, cross-linkage usually occurs by direct peptide linkage of the amino group of diaminophilic acid in the carboxyl group of the terminal D-alanine. In gram-positive bacteria, cross-linkage is usually by peptide interbridge. The kinds and number of cross-linking amino acids varies from organism to organism. In gram-negative bacteria such as E. coli, cross-linkage usually occurs by direct peptide. We can see a direct peptide linkage here, NAM to NAM, of the amino group of diaminophenolic acid to the carboxyl group of the terminal D-alanine. The peptidoglycan chain connected through short tetrapeptide and pentaglycine bridge. The polysaccharide chain consisting of alternate residues of amino acid N-acetylglucosamine, that is NAG, and the N-acetylmuramic acid NAM, linked by beta 1 4 glycosidic linkage. Each NAM residue carries a short peptide chain of 4 amino acid residues. That is tetrapeptide, which has got L-alanine, D-glutamic acid, L-lysine, and D-alanine. The peptidoglycan synthesis. So how it is synthesis? That's what we are going to see here. The N-acetylglucosamine, that is GLIC-NAC, you can see here, plus there is a phosphate group which is added to a UTP, which is a reverse reaction, is to form UDP plus GLIC-NAC plus PPI, that is a phosphate group. And then the next reaction undergoes, in which the PEP comes inside, and NADPH is transformed into NADP plus. So second process is UDP, that is GLICNAC, reacts with PEP to form UDP more to AC, that is UDP muramic to AC. And then the three, or the we call this the 4 amino acid which is being linked, that is L-alanine, D-glutamate, L-lysine, and D-alanine and D-alanine linkage. The 5 amino acids are then added. You see here 1, 2, 3, 4, 5 amino acids are added, and then the next reaction happens. Then what happens in the second reaction? The prompt is the Mur to AC pentapeptide moiety of then is transferred from UDP to dolicol. Another glycnac is added to this molecule. Then the 5-glycine is added once again. You can see in the sixth process, the 5L-glycine is added in the lyse residues of pentapeptide. In the third stage of this whole linkage, the whole disaccharide peptide, especially in the seventh stage, the decapeptide is added to the non-reducing, the decapeptide, that is a primer that has been added to the non-reducing end of the existing peptidoglycan molecule. And then in the fourth process, the peptidoglycan synthesis, we can see that transpeptidase catalase, that is the catalase is used as a transpeptidation reaction to cross-link adjacent polysaccharide chains, especially like penicillins are reacted. Now, about the diversity of peptidoglycan. Peptidoglycan is present only in bacteria. N-acetylmuramic acid, that is NAM, and mesodiaminophilic acid are never found in cell walls of archaea or eukaryotes. In peptidoglycan, the glycan portion is uniform with only the sugar NAG and NAM which is being present, and these sugars are always connected in beta 1,4 linkage. Tetrapeptide of the repeating unit shows major variation in only one amino acid, the lysine diaminophilic acid alteration. More than 100 different peptidoglycan types are known, and the greater variation among them occurs in interbridge. Peptidoglycan mainly consists of 70 to 80% of the cell wall in gram-positive bacteria. Lipid component is little, and in gram-negative bacteria, you see it as 10 to 20% is formed of peptidoglycan, and the lipid content is 20 to 30 percentage. And thank you. So with this, I conclude the video.